Today we'll be discussing reproduction in Welwitschia. Welwitschia is dioecious, that is, two sexes are present in separate individuals. The inflorescence is developed from a series of several transverse ridges arising parallel to the leaf bases. The branching in inflorescence is dichasial and each branch ends in an attractive cone. Several brackets or cone scales arranged in opposite decussate manner are present in each cone because of crimson or scarlet color. Mature cones of Welwitschia provide a beautiful look. So first we'll start with the male strobilus and male flower. Microsporangiate strobilus or male cone is a compound structure having quadrangular cone axils. Just look at this first diagram. This is a male cone. Male cone possesses several brackets, also called cone scales, which are arranged in opposite and decussate manners. Each male flower is present in the axil of each subtending bracket, and each male flower possesses two lateral brackets. Just look at this second diagram. This is a male flower, and these are two lateral brackets. Then male flower also contain a perianth. This is perianth, which is formed from two bracket-like anterior posteriorly placed structures inner to the perianth. The inner to the perianth is present a word of 6 microsporangiophore. So these are microsporangiophore, and which remain fused at the base and form a cup-like structure, and at the top of each microsporangiophore is present a synangium. So these are synangia. So this is synangium. Each synangium is formed by fusion of 3 microsporangia. Each microsporangium contains many pollen grains which are shed through a vertical slit, and pollination is effected either by wind or by insect. With male gametophyte, pollen grain matures to form a 3-celled male gametophyte. But there is not much information about the process of development of male gametophyte. So this 3-celled structure includes a tube nucleus, a sterile cell, and spermatogenous cells. The sterile cells usually gets aborted even before pollination, and spermatogenous cells gives rise to male nuclei or sperm nuclei. So next is female strobilus and female flower. Female strobilus, known as ovulate or microsporangiate strobilus or aviliferous cone, is also a compound structure as seen in male strobilus. Just look at this first diagram. This is female cone. So several decussate brackets or cone scales are present on the axis of female strobilus. So these are aviliferous scales. In the axil of bracket is present a female flower. Inside the subtending bracket of each female flower are present two small lateral brackets, then two envelopes and a single nucellus. Inner envelope function as true integument and prolongs in the form of long tubular micropyle, and the outer envelope develop from two posterior-anterior primordia which fuse with each other in early stages of the development, and this fusion product is also called a perianth. So just look at the second diagram. This is perianth, this is subtending bracket, and this is perianth. Then which this perianth expands into a broad wing-like structure, and this wing-like structure in the mature seed. Then female gametophyte, just look at the second diagram. A single megaspore mother cell develops deep in the nucellar tissues. Diploid nucleus of megaspore mother cell undergoes meiosis, but no cross-wall formation occurs. Also, a linear tetrad is not formed, and development of tetrasporic female prothallus occurs directly. So haploid nuclei of young female gametophyte divide and redivide several times mitotically, but again no cross-wall formation occurs, These divisions result in formation of thousands of free nuclei, as you can see here, these free nuclei. So this is female gametophyte, and these are nucellar cells, and these are free nuclei. Now, formation of cell wall begin irregularly, and each cell contain varying number of nuclei. Many cells, the nuclei fuse to form polyploid, and formation of archegonia has not been reported in Welwitschia. Process of fertilization begins with elongation of pollen tube, which grow downward through the nucellus. Simultaneously, apical cell of the female prothallus also elongate and form prothallal tube, which grow upward. Hence, pollen tube and prothallal tube grow in opposite direction, come in contact near nucellar cap. So, wall between the two tubes get dissolved. Male and female nuclei fuse to form the fusion nucleus. The diploid zygotic nucleus divides and forms two-celled proembryo. The upper cell of this proembryo develops into a primary suspensor, while the lower cell divides and redivides to form a large number of secondary suspensor cells and multicellular embryo. A cap of about 8 cap cells protect the young multicellular embryo, which is pushed down into the prothallus by the elongating suspensor. Welwitschia exhibits a high degree of polyembryony because many zygote and young embryo are produced. However, only one embryo finally matures into seed, and a lateral finger-like process is called feeder is present in the mature embryo. And the winged outer envelope of ovule mature into papery structure, and the seed germination is apogee, and both the permanent leaves appear soon after the germination, and the cotyledons grow for about 6 months and finally die. Thank you.